A surprising connection between the herpes simplex virus type 1, HSV-1, commonly known as the cold sore virus, and Alzheimer's disease has been uncovered through decades of research. While many people have contracted the virus in childhood and live with it in a dormant state for the rest of their lives, this seemingly innocuous virus may be quietly contributing to the development of Alzheimer's disease, particularly in individuals with specific genetic risk factors. The journey began over 30 years ago when my colleagues and I discovered that HSV-1 could reside in the brains of older individuals. This was groundbreaking because until then, it was widely believed that the brain was immune to infections due to the protective blood-brain barrier. What we found, however, was that the cold sore virus was able to bypass this barrier, lying dormant in the nervous system. It can be triggered by factors such as stress or illness, causing cold sores in some people, but can also make its way to the brain, where it potentially wreaks havoc. Our subsequent research showed that people who carry a particular version of the APOE-e4 gene, a gene known to increase the risk of Alzheimer's, and who also had been infected with the herpes virus, faced a much higher risk of developing the disease. This discovery was crucial because it linked a common virus, typically associated with cold sores, to the development of a debilitating neurological condition. We then turned our attention to how HSV-1 might contribute to the buildup of amyloid plaques and tau tangles, two abnormal protein structures that are hallmarks of Alzheimer's disease. In laboratory experiments, we found that brain cells infected with the virus produced the same amyloid and tau proteins that are observed in Alzheimer's patients. Over time, as the immune system weakens with age, the dormant virus could reactivate in the brain. This reactivation leads to brain cell damage and inflammation, which over time could accumulate and contribute to the cognitive decline seen in Alzheimer's disease. Even more startling was our discovery of the virus's DNA within the amyloid plaques in the brains of Alzheimer's patients. This strongly suggested that the virus plays an active role in the disease. Encouragingly, we also found that antiviral treatments seem to reduce this damage in the lab, suggesting a possible therapeutic pathway for slowing or even preventing the disease in the future. Large population studies have further supported these findings, indicating that severe HSV-1 infections are strongly linked to Alzheimer's. These studies also showed that antiviral treatments, when administered during outbreaks of the virus, could significantly reduce the risk of Alzheimer's. This raises an intriguing possibility that the use of antiviral medications might play a role in mitigating the effects of the virus and ultimately reducing the incidence of Alzheimer's in certain populations. Our research did not stop with HSV-1. We began to wonder if other viruses that lie dormant in the body, such as the varicella-zoster virus, which causes chickenpox and shingles, could also play a role in the development of Alzheimer's. When we looked at health records from hundreds of thousands of people in the UK, we observed a subtle yet important trend. Individuals who had experienced shingles had a slightly increased risk of developing dementia. However, those who received the shingles vaccine appeared to have a lower risk of dementia, which further reinforced our hypothesis that preventing infections might reduce the risk of Alzheimer's. Similar results were seen in a Stanford University study, lending further credibility to the idea that vaccines and antiviral treatments could provide protection. One key element in our research was exploring how infections and brain injuries might trigger the reactivation of dormant viruses like HSV-1. We created a 3D model of the brain, incorporating a dormant herpes infection, and simulated both infections and brain injuries. The results were striking. When the virus was reactivated, it caused the kind of damage to brain cells that is characteristic of Alzheimer's disease. However, when we used anti-inflammatory treatments to prevent the reactivation of the virus, the brain damage did not occur. This suggested that managing inflammation could help keep the virus dormant and prevent the development of Alzheimer's. This body of research is a game changer in how we think about Alzheimer's. What was once considered a purely genetic and age-related disease might have a more complex cause, one that is potentially preventable. The possibility that a common virus, often associated with cold sores, could be a significant contributor to Alzheimer's, opens up exciting new avenues for prevention. If we can find ways to keep the virus dormant, either through vaccines or antiviral treatments, we might be able to reduce the risk of Alzheimer's, especially in individuals with higher genetic susceptibility. While more research is needed to fully understand the virus-Alzheimer's connection and how best to intervene, the results so far are promising. This could ultimately lead to new preventive strategies and treatments that target viral reactivation in the brain. What began as a simple observation about cold sores and memory loss has evolved into a broader understanding of how infections could play a crucial role in one of the most devastating diseases of our time. If this hypothesis proves correct, it could change the landscape of Alzheimer's treatment and prevention, offering hope to millions around the world. However, it's important to be cautious as we continue to explore these connections, as we still need more evidence to determine the best course of action for clinical application.